All right, so this is our second meetup here for the joint innovation projects funded through the Build Back Better grant led by the Alaska Mariculture Cluster and Southeast Conference. And we have a few presentations here today. We're representing both the JIP2 and JIP3 cohorts. So it's a mix of research and development projects. And the first project we'll start off with today is with Austria Marine and Kyle Newman. I'll just start with saying that we'll have about, in between 5 to 7, up to 10 minutes of presentations, followed by Q&A for each project. And then at the end, we'll have a more broad discussion around industry needs. We have a little bit of buffer time, so if presentations go a little long, that's fine, but I will try to keep everyone timely if we go over 10 minutes. So yeah, I see you up and ready, Kyle. So I'll just give a quick introduction. Kyle Newman is with Austria Marine, and he'll be presenting on the Planktoscope, a low-cost automated plankton imaging and analysis tool project. You've had a couple of projects, but you can go ahead and begin.
Yeah, thanks, Alex. Yeah, so this project was the goal of this project was to evaluate this new tool that was developed by a group in France in collaboration with Stanford University. Essentially, it's a low cost automated plankton imaging tool. If you're familiar with a flow cytobot, it's similar to that, except that it's not field deployable. It operates on a bench. But essentially, what it does is you take a water sample, you fix the water sample, put some sort of preservative in it, and then which kills and preserves the cells. And you put it into this device, and it is a flow through camera system with a single cell flow path. So it pumps the water sample through a flow cell that separates individual plankton cells, so that you're only capturing one at a time, and then it images all of the cells that are going past. And then the goal there, the stated goal of the group that developed this was it could be an automated plankton identification tool developed for research. And so if you take enough images, you build an image library, and then you can train sort of an AI image analysis engine to then automatically ID plankton in your samples. That's the goal. um, as these things go, it's often more complicated than it seems. And so a big part of what we were looking at was, could this be a viable tool, not just for researchers who have PhDs and have been working in this field for a long time? Could this be a viable tool for oyster farmers or harvesters or citizen science groups, or even research groups in Alaska? Because it's an imaging flow cytobot is probably $100,000, $150,000. These are $5,000, and they sit on the bench. So it's sort of within the range of it's accessible. It's way more accessible. As always with these sorts of trials, our first question is can you figure out how to use it? Can you put it on a bench and get it up and running in a short period of time and produce reliable results with it? And in the case of the planktoscopes, we found that they work really, really well at imaging plankton. We can get great images. The really difficult part is the automated image analysis. Um, I was working on it. I had another friend who um, got doged from NOAA, and he started working on it too. He's an expert in in scientific, research equipment. He's a chemist. He was working on it. I had another person working on it. We were all working on this and trying to figure it out. And in the end, we just found it really difficult to do the automated image analysis. So, the results are kind of mixed, right? I mean, it's really good. It's a really good tool if you want to image plankton. It's very cheap for what it does. It's really capable as a plankton imaging tool. Not good at automated plankton ID. With with the goal of like, could an oyster farmer be collecting water samples on the regular and putting into this machine and get some indication of an Alexandrium bloom coming on with the presence of Alexandrium and the numbers of Alexandrium going up. And I just, with a lot of work, maybe it's doable and a lot of time, but that's not something that oyster farmers tend to have. So, what we're doing now with the planktoscopes is we're getting them out into the hands of researchers who are running imaging flow cytobots or like at NOAA, where they have their own cultures of Alexandrium and other phytoplankton, so they can use them as imaging tools and as sort of digital microscopes to look at these plankton cells. But and we're sort of evaluating how they'll work in that sense. We've pivoted a bit or expanded the project a bit to incorporate two other research tools to these other tools. One is a PSP testing assay that's pretty affordable from a company called SensorReal. It's relatively new technology where we propose to test it out to see if it was effective in Alaska to measure PSP in oysters as well as harvested shellfish species like butter clams and some other species. So we had collected a ton of samples last summer here in Juneau of oysters, and we were going to test them with the sensorial, but unfortunately, or fortunately for the farmers, unfortunately for us, there wasn't really a bloom of Alexandrium in Juneau that led to closures, so we didn't really get hot samples. We've now partnered with John Harley at UAF, and he has a ton of samples saved in a freezer that he knows are hot. And so now we have known hot samples that we can test this equipment with. And we are also talking with BioLoom, which is a company here in Juneau that's developing a bioconductivity, it's not conductivity, anyway, they're developing a technology of their own that could be used for PSP testing. And we're talking with them about incorporating them in this trial as well. We're having a grad student from University of Alaska is picking up this project in terms of the analysis. She's moving to Juneau next month, and she's going to jump right in and test these two technologies against, the HPLC samples from the state or from the Sitka tribe. So we'll have a really good idea, validation of these tools as PSP measurements very soon. And if anyone wants to play around with a planktoscope, I have like 3 of them. And if you have an idea for how, imaging plankton, you want to get great images. It does a really good job of imaging them. You get a whole image library of like hundreds and hundreds of pictures of plankton. It does a great job at that. So if you have a project where you want to take pictures of plankton and look at them, hit me up. I'm happy to loan out one of these planktoscopes.
Thanks for sharing, Kyle. If anyone has any questions, feel free to raise your hand, or you can also input questions into the chat.
Hey, I've got a question, Kyle.
Yeah.
I hope it's all right if I just hop in there. But I'm curious about the financial benefit to oyster farmers and how much of an advantage, if these alternative methods do work out, what would that actually look like to the benefit of farmers?
Yeah. So, um, none of these tools as of now will replace state testing. I mean, they have to be tested by a certified lab in order to be sold anyway. So what they would do is give farmers an early warning tool. Um, what we were hoping with the planktoscope, and there's one part of it that I didn't mention that I think is still a viable option that we'd like to explore with the planktoscopes is if you can image the plankton and you see a bloom kind of coming on, you see the population of Alexandrium increasing, or another one is you start to see uh, larval stages of biofouling organisms increase because they have planktonic stages before they settle. Barnacles and mussels, other fouling organisms have these planktonic stages. If you can start to see that this bloom is coming on, then you could harvest, if if a PSP causing organism, you could harvest before the bloom peaks. Um, if it's a biofouling organism, maybe you could pull that gear temporarily or drop it to a deeper depth temporarily, somehow try and avoid the fouling. Um, the uh, the interesting thing, the other sort of aspect of this is a little challenging, is that there's been a ton of research into Alexandrium blooms and other PSP causing organisms, and it's really variable. You can have a huge bloom of Alexandrium, you can have a ton of them out there, but they may not be producing the toxins. And then you could have a really small bloom, not a lot of them out there, and then you might get really hot uh, oysters. And so there are folks working on this interaction between, because it's not just there's a ton of Alexandrium, so you're going to have a ton of PSP. There's something else going on, and nobody has yet been able to pinpoint what exactly that is. It's not oceanographic conditions, it's not weather, it's not. Um, there's been some great studies on it, and everyone's kind of been like, oh, I don't know. There's a really interesting idea. I'm kind of getting in the weeds here, but there's a really interesting idea that maybe it's zooplankton presence, and the zooplankton are feeding on the phytoplankton, and it's a prey response that they're developing these secondary metabolites as toxins. In any case, if that were to be true, then you could use a planktoscope to see, oh, do we have a presence of these zooplankton and Alexandrium, right? And it would we're trying to come up with these early warning systems so that farmers are not caught off guard by a bloom. The other thing, the more direct testing, so like the sensorial is a tissue assay. You grind up tissue and you actually measure the tissue. If uh and it's a semi-quantitative, so it tells you like basically are you hot or not. Um, if an oyster farmer had a closure, for example, and they already knew that they were not um they couldn't be selling their oysters, rather than continuing to send out tests to a state lab, which is relatively costly. It takes a little bit of a turnaround time. This sensorial gives you a result in 5 minutes. And it's a relatively inexpensive tool. The reader's a couple $1,000, and then it has like some consumables. But the farmer could be testing themselves during a closure and be like, "We're still hot. We're still hot. We're still hot. We're still hot. Okay, the sensorial says we're not hot. Now let's send it to the lab, and we'll be able to." So it kind of saves some of that effort, some of the cost of sending it to a state lab when you know that you're hot.
Yeah.
Um, could also be really useful for harvesters. Again, I don't want this project to say like this is perfectly safe and you can rely on this and go out and harvest or to with abandon and you're going to be fine if you use this tool, but it's better than nothing, right? We basically, harvesters, people who are wild harvesting, have nothing right now. I mean, I guess they can send to the state lab, but it's pretty expensive. So if they had one of these tools locally, maybe you could get faster results. That was
Right.
sort of our ideas there.
Thank you so much, Kyle. I think for the sake
Yeah.
of time, we're gonna have to move on. But
Sure.
really interesting, and thanks for the more in-depth explanations
Yeah.
today. Yeah, it'll be interesting to see if we can find out why these blooms are happening. I didn't know all of that, so yeah, that's super interesting. Okay, so next up we have Elizabeth. Hi, Elizabeth, and she has her presentation already. She's with the Organized Village of Cake, and are Stephen and Justin here as well, or will you be presenting, Elizabeth?
Yeah, good morning. I know that Stephen was going to try to join if he has time. Justin, I don't think is available this morning.
Okay.
So I'm happy to present to everyone some of the questions people might have. I can't necessarily answer because I don't do the field work most of the time. So I can also pass stuff along if Stephen doesn't make it before the end. So my name's Elizabeth Figgis. What was that?
I'm right here, Elizabeth.
Oh,
Stephen.
hi, Stephen. Great. Wonderful. We just got started. So my name is Elizabeth Figgis. I work as a contractor and consultant with Organized Village of Cake, among other things. Stephen Demmer is also on the call. He is a staff member at Organized Village of Cake and has done, I think, most of the field events listed in this presentation, only not the ones that took place before he was hired last year. So we have been lucky enough as a team to be able to get I think it was the round 2 of the innovation project grants. And the very wordy title of this was about developing increased survival techniques for 2 species of shellfish, specifically through trying to implement predator protection. We ended up actually changing the species, but the primary goal of this remained the same. The whole premise of this is that sea otter populations are imminently and acutely threatening shellfish populations in Southeast Alaska. As we have repeatedly seen from when our colleagues, Jeff and others, come down from Seward, from APMI to visit Cake, they do still kind of just ogle and get really excited about the abundance near Cake of the different shellfish species when compared with areas further north that they tend to work in. So there is still quite a bit to be saved, I would say, or to be protected. There are still harvestable populations of butter clams and little neck clams, and to a lesser extent cockles right near the community of Cake. And so there's really this urgent call for the past few years to figure out how to provide physical protection of the beaches people like to harvest at, as well as trying to then add in some amount of enhancement or supplemental seeding of shellfish. So this kind of looks across the arc of 2024 to 2026, but Organized Village of Cake has really started in earnest trying to do various aspects of shellfish gardening and basically just enhancing the beaches through physical raking and tending the beaches since 2022. And even before that, they did join the Southeast Alaska Tribal Ocean Research Program that does do things like PSP testing for folks. So there's already kind of a big net of stuff that this is happening within. But this specific project had 3 objectives. We worked with APMI as they refined their techniques. We were originally going to be looking at soft shell clams and cockles. Soft shell clams are not abundant near Cake, and they're not really consumed. They were not seen as a priority by locals. And so instead, at the start of the project, we shifted and decided to collect cockles, butter clams, and little neck clams for the study. APMI was generous enough to go ahead and seed or reproduce all 3 species instead of 2. And then we really wanted to look at what are the optimum installation and tending techniques for what we're calling predator control blankets, essentially just netting to put down on the beaches after seeding out juvenile shellfish, spat. And then we had this goal to do a controlled 12-month grow out scenario, which I'll get into in a minute. But essentially, APMI just did a great job working on in their hatchery for us, working on getting a whole bunch of seed of 3 species instead of the originally planned 2. One thing that was really cool, they had really strong production numbers for all of their hatchery work. And what they ended up doing in this project, in the 2 years of the work, they developed a technique of sending out 2 different rounds of seed. So in the fall time, they sent out younger, less developed spat, and with this expectation there would be more mortality, but it allowed the teams to experiment and have, instead of just 2 rounds of seeding, able to have 4, which really added a lot of value just to learning about what the options are on different beaches near Cake. APMI also took time to visit sites to come down to Cake twice during the project, once in 25 and once this year in 2026. And that was extremely helpful because prior to their visit, we had all just sort of been talking about things in each person's context, right? So the beaches they're used to up closer to Seward, and then folks in Cake, the beaches that they're used to in Cake. So that just, objective 1 was just kind of hit out of the park by Jeff and APMI, and I can't take any responsibility for that. But then the other 2 objectives are really about trying to work on figuring out how to install predator controlled blanket nets after seeding, and see how that works. Like at the beginning of the project, it was APMI that recommended getting these pre-made nets that we could simply secure to the beach using rebar and rocks as pictured here. We really were not sure how they would hold up. Some of the beaches are directly in front of offices and houses that are shown on the photos on this slide. Some of the beaches are only accessible by boat, and so sometimes are only reached once or twice a year. So a big part of this was just saying, okay, like what happens when we try to put these nets out there? Do they become trashed, and then do they become pollution, or are they something that we can actually work with and maybe scale up? In the bottom right of this picture, the net on the bottom right where there's rocks, that was the first net that was installed, and then the picture is showing people adding a second one next to it years later. And some of the beaches have been incredibly stable. Most of them have. So we've found that by and large, the nets remain undisturbed. Humans are not that concerned or curious enough to rip them up and take them. Animals are not really able to harm them. So we have had proof of sea otters trying to dig through a net and then giving up rather than ripping it. And it slows down things like starfish and crabs significantly enough to reduce all activity underneath them, which is one of our goals. We did find that at one location, the weather just simply destroyed the first installation. And I'll show a picture of that. We found that the nets do quite a productive job of growing algae and kelp passively, which is not that surprising. But what was interesting is that the different beaches were supporting different types of species. And I can explain a little bit of that on the other slide. Now we're at a point where, because we took pretty small nets, 15 by 50 feet. The picture on the bottom right here shows one of them zip tied together so that it's a 30 by 50. They're really easy to manipulate, very easy to move around when they're in either just 1 or 2 of them zip tied together. We are still scratching our heads to try to figure out methods for potentially increasing installations. And right now we're thinking probably keeping them in only groups of 2 or 3, but then just spreading them more across beaches where they're working well. This is also an issue for future discussion and planning about how to make sure that the nets that are protecting shellfish are not also hindering people who want to go out and harvest. So the second objective is actually just also part of the third objective, which is we wanted to have a whole year, 12 months, to look at least 4 different scenarios for what it's like to do grow out after seeding. And since we had 3 species, we had a bunch of different beaches we were working at, we wanted to prepare some suitable habitat, install netting, then use placing seed in cages or bags, and then also try out just sort of free broadcasting, like spreading seed on the beach and then spreading it openly under the netting, trying different ways of doing this. How this actually played out, we ended up actually having 5 different beaches during the project period instead of 4, which was great. And I would say kind of 3 main issues. So this first area is right near the OVK tribal offices. They own property for the offices that's right on the beach. They had back in 2022 started an experimental pilot shellfish garden, which is shown from this aerial photo from above, just a small rock wall that's just like 100 feet long. And it was right behind that rock wall that they did the first installations of predator nets. This is just a really great area. It's right next to the offices, so Stephen and others can look at this on a daily basis and kind of monitor and do work. One thing, though, that we noticed, which is shown in the picture on the left, this year, after looking for 2 years watching how the nets behave, it really seems possible that this location, because there is a small fish net pen from the local hatchery, it really seems like there might be a little bit of eutrophication or too much nitrogen perhaps from the fish feed that's going into the water there, because what's happening is instead of luscious, healthy kelps growing, we're getting this more mucky green algae, which we're concerned might actually be suffocating some of the buried seed, and especially things that we put over the nets that then kind of add additional coverage. So we're trying to figure that out, if this is just kind of not the best place for expanding the nets. But further out of town, but at one of the most heavily used beaches, like for the culture camp that they host and for personal harvest, is their Point White Junior Beach. We installed one of the second set of nets, so one of the first ones we ever installed back in November of 2024. And this has been a really exciting area to see on the right evidence of an otter trying to dig through this net and giving up and moving right next to it. On the left, there are visible otter pits all around this net. And this net, just over a few months of time, grew the most exciting luscious kelp that our visitors from APMI were really excited about, how much it probably will help the shellfish underneath it. I'm going to just run through the other scenarios really quickly because I know we need to move, Alex.
Yeah, I was just
you guys
if you
can
could
all
wrap
take
up
a
in the
look.
next minute or two, that would be great. Thanks.
So this one's really exciting for a lot of reasons. It's happily growing kelps. It looks like a really great way to protect the shellfish, as our goal was. Other beaches completely destroyed by weather. We really need to go back to the drawing board because there are some beaches like this one that the community really cares about, but we have not figured out how to protect yet. Stephen prepared some really nice information about what they most recently did this summer with the final seeding event, using different types of burying mesh bags with spat and free broadcasting. And then there's just been a huge amount of community interest in this work. This is just one of the seeding events this summer. And Stephen's done a really great job at getting folks involved in everything from what they call fluffing or even feeding the clams underneath the netting, to just general installation and tending. We have not really had to repair much in the nets. They've done a ton of events over the past 2 years. The biggest challenge we've had are being able to really measure this through biomass surveys because they're super expensive to actually get folks out and do a controlled survey, especially now that we have 5 locations. I would say the biggest thing we're looking forward to is using UV light to try to review the latest seed, because APMI was able to do marking of the last round that went in. So next year we're hoping to be able to actually more quantitatively see, like, are these young shellfish actually from the project. we're going to be looking for more funding because we feel like we don't really have a huge amount of quantitative data on this yet. We have made a lot of mistakes of how to kill seed and shellfish, and we've had really good luck with the nets. But yeah, thank you, Alex. Sorry for going over time.
No, thank you so much. It's great to see the pictures, and I love seeing how much community engagement there's been as well. That's amazing. I might direct questions to the chat. If anyone has questions for Elizabeth, please go ahead and enter them into the chat, and we can probably move right on just to make sure that we can fit everyone in here. But we could have time for one question if anyone has something, or yeah, if just go ahead into the chat might be best too. And then Cameron, if you are ready to share, and pull up here.
Hey, yeah, I can share. I say that, but every time I try to share, it doesn't work out. That's why I sent you.
Yeah,
I know, that's why I sent that. Is it working?
Yeah, looks great.
Wow. All right, there we go. Well, hey everyone. My name's Cameron. I'm here up in Seward at the Alutiiq Pride Marine Institute, which you just heard quite a lot about. It's been great to hear so much praise about the shellfish work. Of course, I'm on the kelp side. And specifically, I've been working on gametophyte techniques for the commercial kelp hatchery efforts. So this was the first project, the GIP project, the first funding that we got to actually establish a gametophyte program here at APMI. So I'll just kind of take you through the work that we've done. So just a little bit of background, the gametophyte stage is the sexually reproductive stage of kelp. So, you know, a lot of people are familiar with the typical hatchery process where you go out and collect source tissue, release these zoospores here. Those will turn into gametophytes, both male and female, which then sexually reproduce, and you have a new round of kelp that is then outplanted. And traditionally, this has been done just with the manipulation of the source and zoospores. And our goal is to basically culture the gametophytes independently and then use those to control the outplant timing and the reproduction on both seed spools and what they call direct seeding in different practices that have various benefits and commercial applications for kelp farmers. And so we've been doing this kind of on a broad scale through Alaska-based protocols and best practices and through equipment accumulation so that we can, you know, get the best product we can as the gametophyte style of hatchery is a little bit more involved and technically demanding. So our JIP project had two main objectives. One, to optimize the gametophyte husbandry protocols for sugar kelp in Alaska, and two, to establish direct seeding protocols to achieve optimum seeding efficiency and consistent sporophyte density. So yeah, these are, honestly, looking back on these are pretty lofty and very strong objectives. But we've learned a lot, certainly throughout the process. I'll kind of take you through just some general ideas of what we were doing. So when I got here, we didn't have a gametophyte program. I kind of just basically had an empty Connex, you know, with some benches, and was like, all right, we're going to start doing this. We're going to start collecting gametophytes and culturing them. So honestly, the most cost-effective way to do that, if you don't have a lot of money for, say, a $40,000 lab incubator, you can use wine coolers. And so that's what we were doing. Use some, you know, DIY light installations. And we're still using these, and they work pretty well. In this photo here, on the top, we have like a bunch of isolate cultures here. So these are all very independent cultures where you have, you know, independent male and female gametophytes in their independent little sections. And down here, you have a mixed sex culture, which is just basically what happens when you release zoospores and then have all of them grow up as gametophytes together. This right here, so like, I'll take you through the process. once we started growing these cultures, really kind of when it came down to measuring the biomass, this is where we kind of got stuck because gametophytes grow very slowly, and it's tough to get enough biomass to actually measure the biomass precisely, because the best way to do is using dry weight measurements, which is destructive. And when when you have a lot of objectives and plans for this, these live organisms, it's tough. And so what we ended up doing is basically calibrating a volumetric measurement using these PCV little capsules here, which you put in a centrifuge, and then you can measure just like the pure volume of the biomass in a particular sample. And then, once you've calibrated that to your dry weight samples, then you can use that. And so that's kind of what we ended up going with. So we ran an experiment based on objective one, which I think many of you have seen before. I've presented on these results before. Essentially, we just looked at how density affects the specific growth rate in cultures. So we had a mixed sex stock here, and then I've diluted those down to various densities here. You can kind of see the opaqueness changing. And then we put those on this orbital mixer just to kind of stimulate water flow, because in a larger culture system, you would have it's common to have bubbling or mixing of some kind to increase growth rates and ensure even light distribution and nutrient flux into the gametophytes. And so we tried to stimulate that as best we could, basically with this plate here, which would just basically shake the water inside of these cell plates while also maintaining, individual replicants, which you can't do in a large system. And naturally, what we found is you end up with, slower growth at higher densities, really tapering off pretty consistently at 0.17 milligrams per milliliter dry weight. And that makes a lot of sense. The other thing we were looking at was sexual reproduction and sort of how that's affected, because if you're growing mixed sex cultures, you really don't want them to reproduce before they're ready. And we did find that sexual reproduction was repressed after 0.04 milligrams per milliliter dry weight, which is actually like not a very dense culture. So that was interesting. So the ultimate goal here, right, was to use these larger scale production systems. So thanks to the Southeast Conference, we've been funded these and have acquired now 2 racks of these, which are the industrial plankton seaweed bioreactors. And these are specifically made for gametophytes. And basically you have each of these chambers as an individual growth chamber where you have CO2, you can control, air influx, light, temperature, photoperiod. They're really advanced machines, and they're made to be completely sealed. So like once you start a stock in there, you can exchange nutrients, you can harvest without ever having to expose that stock to potential contamination from the air. So, it's a very biosecure system, assuming that the initial stock that you put in there is biosecure, which it hasn't been ever once for me, because we've been dealing with mixed stocks, and yeah. As for objective 2, which I was really excited about, we were going to make this wave tank to test the settlement of gametophytes with a direct seeding method. So instead of using this, twine seeding as like an intermediate, you can go and take your gametophytes and put them straight on lines in the ocean, which is a really popular idea amongst farmers or anyone who's ever had to deal with seed string. But it is also kind of tricky because, kelp, you're bypassing a pretty big stage in the kelp's life cycle and how they, biologically attach to substrate. And so you end up losing a lot of them to the water. I've been told it's typically around 95% loss with direct seeding is what you should expect. And so even like small increases in that number could pretty drastically reduce the cost of seed if you're able to spread that over a larger area. So we were going to make this wave tank, and unfortunately, we only got about halfway through before we ran out of money. This is a photo of it on the left. We ended up going with this wedge system, and this isn't as far as we got. We kind of built this frame around it and had a electrical actuator on top and reciprocating actuator, and basically the wedge would force down, and that would come across. And the idea is that you can place lines at different depths corresponding to different orbital velocities of the wave pattern as it travels down the water column. So yeah, I'm really hoping to get some internal funds to continue that because I mean we almost have it, and it would be a really cool project. But unfortunately, we don't have any results from that because the wave tank was never actually finished. The second photo here is this is actually a filter disc from one of our larger gamified cultures, and what you see here is a lot of contamination. You have all this green algae in here, along with all these like little brown dots, which are the gametophytes. So pretty high density, but also very unclean. Later on, we did move on with Southeast Conference grant as well, and we did get really success. We had a lot of success with induction using those SBR systems. So that's like combining the male and female gametophytes and then like inducing them to reproduce. So here we could see that you have a lot of these juvenile sporophytes here. Yeah, really nice looking. So, I guess in summary, the GIP program, what we wanted out of this was pretty lofty, and we did not achieve. I would say we did not achieve either of our objectives for this project. But in the grand scheme of things, what this did is it really is set the seed for the program itself. This allowed us to start accumulating resources and equipment, supplies, just kind of working through our own protocols and how to actually grow gametophytes, making connections. And it's definitely snowballed into a much larger project where, we started with the GIP award, then we had some Southeast Conference money, and that allowed us to go out and actually do some direct seeding in the trials in the ocean. And then we got some equipment to actually expand. So now we potentially have everything we need to actually run a commercial gametophyte hatchery. Then, in the spring of this year, the Fish and Game approved the use of gametophytes for commercial purposes. So we're in the process of creating commercial specific gametophyte stocks that can then be used for non-research purposes, which is exciting. And then we've got a pending RAC project that would potentially fund us for quite a number of years moving forward. That would be sort of a hybrid research and commercial program where we're collecting data, but all the gametophytes that we're producing would actually be viable to be sent out to farmers. So yeah, that's kind of the gist of it. Happy to take any questions. Really appreciate you guys listening in.
Thank you so much, Cameron. And it's great to see the timeline too and just the evolution of the work. With is the RAC proposal approved? Are you just in the final
No,
phases
it's
there,
pending. So we made it
okay.
past the pre-proposal stage, and I'll be going
Yeah.
in October to present our proposal, but we'll see how it goes.
Yeah, exciting.
Yeah.
I mean, that's a big step to make it through that phase. So
Certainly.
all right. Do we have any questions and Elizabeth, if you're still here, there's a question for you in the chat.
Cool. 1 second, let me expand this.
And you can just, you can just
Yeah.
respond in the
What
chat
methods have we
there
tried?
if you
So
want.
this is where we're kind of leaving off at. We originally worked with just some rebar, and then that first picture that showed all the rocks, all the boulders around the net, we like really overdid it at the beach in front of the offices. That gets like no significant wave action almost year round. So then we were like a little more bold and just did rebar. And just did rebar to the extent we thought it would help keep animals from going under. So every couple feet, every 2 to 4 feet. But Long Beach, which is where I showed the photos of the tangled nets, just completely ripped them up and rolled them up, as you could see. So we're either considering doing the simple small rocks around the edge there, or doing something a little bit more derived, like screwing anchors in. And then I don't know if the other questions were for us, but yeah, we're kind of just trying to figure out because we want it to be low key enough that it's easy to go out and lift them up and look, and also easy to go up and move their location. So we're just trying to figure out the most parsimonious idea.
Yeah, that makes sense. Okay, thanks.
Yeah, I'm happy to talk to you about that more. I've had really good luck with screw anchors on beaches here to build like oyster farming arrays. That was actually my GIP1 project, was building some intertidal oyster arrays, and the screw anchors have been awesome. So I'm happy to chat with you about how we did it and figure out how we might be able to how you might be able to use screw anchors to secure your nets.
Yeah, that would be great. We might just be trying that this winter then. Somebody had another question asking about interest. Yeah, I mean, I think a lot of people had interest, and APMI has been doing some of this net coverage for years, so they have a lot of experience working on ways that it can help. We have not had anybody reach out directly to us that I know of. I don't know if Stephen's still on here to answer anything about that, if people have reached out.
Thanks,
Reach
Elizabeth.
out, reach out for what, Elizabeth?
Oh, I was just curious, Stephen, if other communities have reached directly out to you or Justin to ask about the seeding and predator netting methods.
No, no, they haven't.
All right, I think we'll move into another question for Cameron from Johnny. What does sending out seed to farmers look like?
Hey, Johnny, that's a good question. I think the two ways we would do it is we would either send out gametophytes to a regional nursery, so basically skip the source in spore release, and then seed spores using the green wave method, but regionally, wherever you like to get your spools from. Alternatively, if you did have access to direct seeding equipment, then we would send the gametophytes directly to the farmers themselves.
And then Abitzer has a question about organic compliance.
Yeah, hi. For the seeding, after it goes through the whole process, and you showed a pretty sophisticated system there. Um, Whatever chemicals are used or sterilizers that are used, how does that play into organic compliance? And can we say that the seeds are organic when we work through organic compliance, especially for foods?
That's also a really interesting question. I've not thought about this. I mean, I've thought about this a little bit in terms of other people's projects, particularly with germanium dioxide and how that impacts organic claims. We have used both GeO2 and kanamycin, which is an antibiotic, in the past. We've stopped using kanamycin, and actually, this rack project that we're going for right now has a pretty large associated microbiota component. So that's in partnership with KelpArc, with the idea being that instead of basically trying to kill off all these, bad contamination, is you can, if you understand what associated bacteria are beneficial for the gametophytes, then you can basically try to both create cultures around that and actually use probiotics to stimulate their growth to then promote the long-term health of the gametophyte stocks. So using that method, I would imagine it's you. I mean, I don't know what the regulatory or the details of what is organic or not, but I imagine moving forward that would be probably a good way to go regarding organic and health claims.
Yeah,
Thanks,
I think the
Cameron.
organic approval authorities, do have a say into what can or cannot be used, and so just awareness of that would be very important going forward as to
Yeah.
what are their requirements and how do you comply,
Mhm.
or can it even, and if it can't comply, then you have to ask for exceptions. So I mean, I'm
Right.
just saying that there needs to be some effort
Yeah, and I think
working with authorities.
that's what happened with germanium dioxide use, because it really is important. I don't think we can really get away from that with diatom control. Um, but um, but again, it's like you don't have to use that all the time. So it's like, you could probably get an exception if you're only using it for the initial inoculations. But yeah, I'll definitely keep that in mind. Thanks.
Great. Yeah, if
Thank
there's
you.
any other questions, we can keep going through the chat, and I'll invite Francis to share your screen. We have Francis with Pacific Hybrid on establishing an oyster broodstock program for the Pacific oyster.
Right. Right. Thank you, Lex. You guys hear me okay?
Yes, can hear you great.
Okay, great. So kind of the overarching goal of this project that we're into our third year of GIP is developing broodstock and also specifically oyster strains that grow and survive better in Alaska waters. As we know, it takes 4 to 5 years of kind of a growth cycle from seed to a market-sized product in Alaska. So if we can kind of speed that up a little bit, it will really benefit the industry as a whole and also individual farms. And kind of a second goal of this project is to as kind of the state is developing its own hatchery capacities, we feel it's important to have a Alaska-based broodstock repository because for now it's not really permitted to import broodstock directly from out of state into Alaska. So there are two ways that we can get seed onto the farms. One is to produce outside of state and ship that into the state when they're still small, or we can have Alaska kind of we can have broodstock kind of based or housed in Alaska, and then that will allow us to kind of Alaska-based hatchery to produce their own seed. So we've been working toward those both goals. And for the first one, developing broodstock, we have produced 3 different cohorts, total of about 60 hybrid crosses of Pacific oyster. This is the hybrid Pacific oyster, same species, but crosses between different genetic lines. And all 60 were outplanted at 2 different sites. One site you see kind of lower right. One site near Ketchikan, the other side more closer to Juneau. And so this year we've been evaluating all 3 cohorts, including first cohort with monitored for 3 years since 2020, 2023, 2024. And then GIP2 cohort 2 that have been monitored for 2 years. And then also this newest cohort that was produced in February and then outplanted in June. So all of our plants are kind of mimic the actual commercial production environment, such as what you see in that picture in the lower left, where there's a rack of kind of experimental crosses next to the actual production oysters, commercial production oysters. And we have also, so after we finish the measurements for the first cohort out of GIP1, we started a repository that initially has 10 crosses that perform better than the others. And we expect to expand that repository as we finish monitoring for GIP for cohort 2 and 3. All right. And in terms of the crosses that we did for cohort 3 from this year, was a diallele cross using 7 different inbred lines, genetic lines of oysters. And the way we did it is cross all these lines in all possible combinations. And of the beakers you see in the picture is every beaker contains a specific cross before we put them into tanks. So every time we do a cross, there's usually about 40 to 50 different tanks going at the same time. And we outplant half of the table. So the ones that are highlighted in yellow were the actual crosses that got outplanted in the field. So this way we get to estimate the genetic components for the families that didn't get outplanted because they are related to the ones in yellow. Right. In terms of field growth, this is using cohort 2 as an example. So they were produced back in 2025 last year for GIP2. And then we have measured it 3 times. The first time was when they were just went out of hatchery. And then also after the growing season last year. There was a bit of delay last year because we have to switch site. So the second data point was actually taken in the winter. And then also a third data point this year before we re-outplant these families. So all we saw was a really big range in their growth, in their sizes and growth. As you can see, the top, sort of the highest, the family that weighed most had about 27-fold increase in weight, as opposed to compared to a 9-fold increase. So that's like a 3-time difference in their growth potential. And then going to a quick summary. So we are and also kind of have developed a 3-year monitoring protocol because the growth cycle is fairly long. What we found out was it really takes 3 years and also 3 cycles of evaluation for before the oyster can get to a size that we feel like we're confident about the data quality. And then as you saw in the last slide, there was a pretty big difference in yield. So that variation will allow us to select for better performing hybrid crosses. And then also one thing I forgot to mention is that because of the approach that we take, we're able to actually go back to the same, we're able to go back to the same genetic lines and produce the same crosses that perform well. So for instance, this year, we used the same 26 line that did really well last year. And so we use that and produce the same crosses again, hopefully to duplicate our results. And so after the 3-year monitoring that I mentioned, and we select higher performing crosses and then put it into a repository for future uses by either research or commercial hatcheries to produce hopefully higher performing oysters for Alaska. And lastly, we have planned our last round of data collection for this JIP next month, hopefully to wrap up JIP cohort 2, and also add one more data point for the newest cohort. And that will be all. Thank you.
Thank you so much, Francis. It's great to see the progress. If anyone has any questions, can go ahead and ask or write into the chat. Francis, can you talk a little bit about the collaborative work with Grow Ketchikan and the hatchery and the equipment grant and how you're going to be involved in that and how that will kind of expand the oysters for Alaska farmers.
So like Alex mentioned, we received as a partner, a equipment grant to develop hatchery and nursery capacity for kelp and for oysters in Ketchikan. So Grow Ketchikan and Sea Grove Oysters are the kind of lead entities on that project. So our role is to help them develop the bivalve component, the oyster component of the hatchery. And because of the testing, one of our testing site is in Ketchikan. So we have a brew stock group there that we're able to hopefully be able to use them to use those group of brew stock and the new hatchery that we're going to develop to produce seed directly for the local industry.
Do you have any other questions from the group?
Yeah, I've got a question, Francis. What would this take to actually become available to oyster farmers? Are there regulatory hurdles? Are there production hurdles?
I think in terms of seed, I think they're available right now because we can import seed into Alaska. So we have 2 hatcheries outside of Alaska, one in Hawaii and one in Manchester, that we do production for farmers. So all of the oysters that we tested stay in Alaska, but we do have their siblings in Washington, in Hawaii, that we can use to produce oysters that are supposed to perform better. So seed are available. So broodstock for production, broodstock in Alaska for production, I think will become available hopefully next year. When they get to this will be from the first cohort. So look at it should get to the reproduction size next year.
And
Great.
does the enhanced seed, would that cost more than normal seed because of the efforts that you guys have put in?
No. I think our kind of, I guess, pricing for now is basically match other commercial hatchery.
Awesome.
Great.
Well, really, really nice work.
Yeah, thank
Yeah,
you.
yeah, it's really exciting. Thank you so much, Francis. All right, we can move on. Kristen.
Excuse me.
You
Good
have your
morning.
video
Hi
here.
there.
Okay, yeah, I'll give you. We have
Yeah.
Kristen Smith here with the Prince William Sound Economic Development District presenting on scaling ambient salted processing to improve supply chains and low-tech preservation methods. Hi, Kristen.
Hey, so do I have permission to share?
You should.
Okay. Let's do that. I do view slideshow. Okie doke.
Perfect.
Yeah, great. Okay. So yes, I am the director of the Prince William Sound Economic Development District. Our office is based in Cordova, and we've been working with the Prince William Sound kelp farmers. And also this past spring, as we were processing, we rented some warehouse space from Silver-Based Seafoods and teamed up with Blue Dot Kitchen to do this, to see if we could scale up salting kelp as a way to help with transport and making it more expedient to ship instead of having to dry right away or freeze, both of which are quite a bit more expensive and obviously more immediate. So yes, whoops, oh no, sorry, what's happening here? I'm just, okay. Yeah, so purchase some equipment in April. It wasn't super equipment intensive. It was pretty, what's the word, low level. We didn't use anything really fancy. Travis Bettinson from Blue Dot Kitchen bought a couple different kinds of totes that he wanted to experiment with, a whole bunch of salt and some agitation blades and a mixer. And we ended up salting about 2,000 pounds of sugar kelp in not even a full 2 days, I think. So maybe it was just 1 day, actually. Yeah, so there was that. And so this is kind of how we did it. We bring the totes with the seaweed in and put them, tip them onto a sorting table. We borrowed this conveyor from Silver-Based Seafoods that had a big plastic belt on it. And yeah, set up a system. I think Lindsay said that she and Taya also marked, they used blue tape on the conveyor belt every 5 feet, and they would allow it, kind of stack the kelp to about 4 inches high, and then they knew that they needed to apply 2 pitchers of salt to every 5 feet of kelp that was stacked that high. So that was kind of the ratio they came up with. And yeah, what else? Yeah, kelp had been harvested by the farmers the previous day, and we stored it overnight in a refrigerator van. Yeah, and I guess Lindsay did note that one of the totes that they salted that had kelp was harvested 2 days before, and they could tell the smell was noticeably different. So we don't know about the microbial levels, but it was not, we ended up, they did talk about using, about the idea of having the kelp in a tote and using some kind of a mechanical mixer, but they realized, I think, that would be a lot heavier to manipulate and wouldn't really work that well. So we settled on this idea of just using the conveyor belt, which was, like I said, pretty low tech, but pretty effective. Yeah, so that's the whole, you can see the whole operation in that. from that angle. And yeah, they came up with a ratio of 30% salt to kelp by weight, seemed to be what was achieving the stabilization condition that they wanted. And Lindsay did talk about how she noticed just how quickly the salt drew the water out of the kelp. Just within minutes, you could see some of the drainage coming off, some greenish liquid, and that was pretty immediate. And so they also had to make sure that the totes could drain a little bit so that all that liquid the kelp wasn't standing in all that liquid. Yeah, and so we had the kelp moved back into the totes after it had been salted. And Blue Dot Kitchen has been sampling once a month for water activity and certain pathogens, which are listed here. Yeah, and so far, the water activity readings are good, but Blue Dot Kitchen says that they have noticed some significant quality changes in the kelp, and that as we've done it so far, they probably wouldn't use it for a high quality food product, but he's going to be testing it again in September just to check on the water activity level and the microbial loads. So yeah, the idea had been to see, could we achieve 6 months of stabilization? And so, yeah, still working on that. I think that's it. Yeah.
Thanks, Kristen. Had Blue Dot been trialing other forms of salt? Like, is this a completely new process for them in general, or
I think it's relatively new. I think they were trying to build on the work that the Kachemak Kelp Hub had been doing over in Homer, Catherine, and Evie. Yeah, I think they were hoping that it would lead to a way to be able to stabilize it right away, you know, in a better condition than you can do with the citric acid. You can't use that for food grade. And so, yeah, they'd been hoping that they could stabilize it right away and ship it down and then do more processing at their facility in Seattle.
Has it been tried against the Kachemak product?
That's a good question. I will ask him.
Yeah, the same question I had, you know, does he find the quality sufficient with the Kachemak Kelp Hub salted kelp?
Yeah.
And it's just something that's tweaking this process, or
right. I will find out and report back.
Abitzer, go ahead.
Yeah, just curious as to what the intended market would be with a salted kelp, so we stabilize it, and then would it be feeding different market streams or just the food market?
I'm pretty sure Blue Dot Kitchen. I know Blue Dot Kitchen was hoping for the food market. That's their sole focus. Really is producing a range of food products, everything from like snack food, salty snack foods. I think ultimately they are hoping to produce some kind of soup and broth mixes, and then also working with some high-end chefs. But yeah, they're pretty exclusively focused on the food market.
Okay. Yeah, no, I think for foods, I kind of understand it. Just wondering if the intention was that it would also preserve it and extend it for other product streams, non-foods. But then the complexity of all that salt being in there would be a huge something to figure out.
Right.
Thank you.
Thank you.
Thank you,
I'll double
Kristen.
check on that, but I'm pretty sure.
All right, I think we can move to Lindsay Olson with Greenwave. Lindsay will be presenting on novel processing of 5 kelp species for cosmetic and nutraceutical products.
Awesome. Thanks, Alex. Hi, everyone. Nice to see you guys. Yeah, it's nice that Kristen and I were next to each other because this work happened in concert with one another. So yeah, Abizer, to your question, this was sort of a parallel processing experiment that happened last spring at the same processing plant where the Blue Dot Kitchen and the salting trials happened. We ran in conjunction with Spencer Sarin at SCRC Consulting a sort of small number of tests on developing extracts for cosmetic and nutraceutical products, or essentially uh, kind of assessing the viability of accessing those markets. So the background context that Kristen kind of just shared was that the Prince William Sound EDD had received multiple pieces of kelp stabilization equipment through an SEC equipment grant in previous years, including a Vincent VS12 shredder, this TSP6 Vincent screw press, and the Serpidus drum dryer. The EDD had leased space from Silver Bay Seafood's meal room for pilot processing activities in May of 2026 and was sort of like a stand-up pilot kelp hub in Cordova this past spring. And to help sort of like coordinate the flow of kelp, food grade kelp that was going through that processing equipment, we worked closely with Kristen and Lindsay at the EDD to sort of coordinate all of the food grade processing activities. And Greenwave ended up purchasing the kelp that went through the food grade processing streams and as an effort to kind of help de-risk some of this equipment and test things out. With this project specifically, we were trying to answer which new market pathways could Cordova access with its new stabilization equipment, and then also what's in the kelp, what's it worth, and what markets will pay for it. Separate to the scope of this project, we also ran a bunch of dryer trials in close coordination with Lindsay Hammer at the EDD, and that work wasn't in the scope of this project, but it was also really getting at those same questions of like, what new market pathways could Cordova access with this stabilization equipment? So the partners on the project, we bought kelp from all three Cordova farms, Royal Ocean Kelp, Noble Ocean Farms, and Wild Blue Mariculture. Worked closely with other Lindsay at the EDD to kind of coordinate all of the flow of these different activities. And then our sort of analysis piece came and consultation came from Spencer Sarin, who was bringing in the sort of chemistry science side of looking at what was in the kelp and what could it be used for. So the four objectives that we started out with for this project were to, one, like document the pilot process. So just create sort of SOPs for the new equipment, figure out like what was the best way to be running this different equipment and how could it be replicated in the future. And then we were interested in using the screw press specifically to make a stable liquid for the cosmetics market. This was Spencer's question of whether or not the extract that came out of the screw press could be suitable for cosmetics. And then also to take the solid fraction of the screw press and get the kind of a breakdown of both minerals and phenols of what's in that solid compound. And then the last objective was to confirm that the facility is ready, sort of do like an overall food safety audit and consultation. We ended up not completing that last one through the scope of this project because separately Greenwave and the EDD worked with Barnacle Foods. We hired their director of people, Jeff Smith, to come up and give a food safety training for all of the farmers. And Barnacle wrote a report with kind of next steps and sort of recommendations for getting the processing line up to food grade standards in the future. So that work happened slightly outside of this scope. So for this project, we were looking at all 5 kelp species that are commercially grown in Cordova. So giant kelp, sugar kelp, bull kelp, ribbon kelp, and split kelp. And all 5 species were dried and tested for compositional analysis. We ran sugar kelp, bull kelp, and split kelp through 3 different process pathways to kind of try and figure out if there were certain tweaks to the process that could unlock new potential in terms of what was in the extract and what was able to be used. The typical run size ended up being around 200 pounds. That seemed to be the minimum amount that was necessary to load the screw press properly to get a full extraction. We've found that with any smaller amount of volume, the screw press wasn't able to press it all the way through, and we weren't able to get a good extraction. So the way of our sort of experimental design and the way that this worked was we took the raw kelp that was landed at the dock. It was like done, a visual inspection was what happened, and it was sort of sorted and cleaned, but no rinse. And then it either went straight into the screw press as just raw kelp, and then there was a liquid fraction and a solid fraction, and I'll talk about how those were stabilized in a second. We also tried another treatment of shredding the raw kelp and then mixing it 2 parts kelp, 1 part boiling water, and sort of agitating it. And the idea here was if you expose the kelp to warm water, does that help break down some of the compounds that will then make the good stuff more easily extracted in the screw press? And then the last treatment was to again, mix it 2 parts kelp, 1 part boiling water, and then add cellulase and citric acid to help stabilize the pH and let it ferment for a week, and then do the same press process. So each treatment or each batch had these 3 treatments. All of the kelp was then screw pressed, and we found that the best sort of in terms of operations, we got the best results if we started at like 15 RPMs to get the screw press going. And then once it had kelp in the auger and was moving it through, we ramped it up to 30 RPMs. This is a video. I gotta mute myself because I say some really embarrassing like commentary. But this is a video of just the sort of the process and the screw press. So right now we're taking a barrel that contains this 2 parts kelp, 1 part water slurry and this is split kelp, which was really gooey, noticeably gooey, and then we poured it into the hopper, and there was some troubleshooting. This like goo that you see coming out the top was a mistake that we had to fix, but it essentially the 2 twin augers, the screw press, push the material through, and the liquid comes out those side bars, and the solid is kind of disposed of at the bottom. So after we had these 3 treatments that went through the screw press, we had then 2 fractions, the liquid fraction and a solid fraction. And the question was that Spencer was really interested in exploring was, could we use sort of like basic, inexpensive stabilization additives that are common in food and cosmetics to keep this liquid fraction shelf stable without refrigeration. So the way that this experiment was designed is we pasteurized the kelp liquid, the extracted liquid. We heated it up to 170 degrees Fahrenheit, just in a kettle. And then we adjusted the pH with a 25% citric acid solution, and then added in 0.2% sodium benzoate and 0.2% potassium sorbate, which are standard cosmetic preservatives. And then we just poured it into bottles, capped, and kept it shelf stable in a cool location until it was tested, and it will continue to be tested as time moves on. And here's a little video of mind-bendingly malleable. This is the split kelp goo. And one of the things that really surprised us was just how gooey the split kelp was. The sugar kelp acted much more like a liquid, but the split kelp had this really stringy consistency, which we'll learn more about from the testing results. With the solid fraction, we used a cabinet dehydrator. The original plan had been to run the solid fraction through the Cirripedis dryer, but at the same time, we were still troubleshooting how to use the Cirripedis dryer. That turned out, and we had very small volumes of these trials, so we ended up just using a cabinet dehydrator to get samples of the solid fraction. So we dehydrated it for 18 hours, crushed it up, and then put it in Ziploc bags with desiccant packets, and then all of those samples for the different treatments, the different batches, the different species were then sent out for lab analysis. So yeah, again, we had 3 farms, 5 species, and liquid solid fractions from each run. And the fermentation treatment happened 7 days after the other two. So we did the first two in the very beginning of our time in the SBS plant, and then we waited a week and did ran the fermented treatment. We sent it out to 3 different labs for testing of different components of the products. And so far, the only one that we have received back is food metrics. So there's still a big kind of question about like the results are inconclusive because we haven't gotten the majority of the data back from the lab analysis. But I can share with you what we know so far. So this is the food metrics lab, which is the only data set that has been sent back, was looking at as to whether or not there was any biological activity in the pressed liquid. So essentially, was anything growing in it? Was it effectively shelf stable? And kind of big line takeaway is that nothing is growing. Every batch tested of all 3 treatments came back clean at the 2-month time point. No pathogens detected and no indicator organisms like no mold or yeast. And everything looks shelf stable. So the kind of big picture takeaway is that this appears to be an effective method for creating a shelf stable extract out of kelp with the equipment on hand in Cordova. So yeah, the data suggests this could be a suitable processing method for cosmetic grade seaweed extract. Said Spencer, I asked him recently what would be different for a food grade to make, could this process also be applied to food grade extracts? And he said, yes, that all of the additives are food safe, but that we used a slightly higher concentration than is typically used in food shelf stable preservation, but he expects that we would have similar results with a slightly lower application, but that hasn't been tested. And it also tells us that this screw press equipment provides a new opportunity for farmers to access markets that are looking for a liquid fraction of their of kelp, and that there's a way for them to create a shelf-stable product without any sort of cold chain logistics. And then, as I mentioned before, the compositional data is still coming, and that will tell us a lot about like what is actually in the extract, and then what potential markets might be interested in that. So in terms of next steps, we're going to continue to monitor the shelf life. We have our 2-month time point. We'll continue for a few more tests. We're going to review the stuff that comes back from the lab, both the liquid and the solid fraction, and then submit our final report and publish open SOPs on how to use the screw press and this process for pasteurization if anybody wants to replicate it.
Thank you, Lindsay.
Yeah.
It's exciting. One quick question with the extracts available on hand, are those going to be sent or worked with by Spencer, or any, like, is there a potential collaboration with cosmetic companies that Greenwave's already working with? Or like what would be the next steps if it goes through all of these processes and things look good?
Yeah, Spencer did retain some samples for that he sent to a potential customer that was interested in just sort of seeing the analysis or seeing the product that could come out of this sort of processing method. So that's definitely like the warm, there's like a warm lead there. And otherwise the volumes that came out of this process were so small, they were just designed for testing. So there isn't like very much additional surplus. There were a couple samples of the split kelp that Amber Morris, who grew the split kelp, sent to a couple of potential customers she has for analysis. But the real goal of this project was just to sort of like try it out, make sure that it worked and it was shelf stable. And so now we know it is going forward for next year if we want to produce more.
Right. Any other last questions?
I have one. I have a question.
Yeah, go ahead.
Yeah.
Lindsay, again, hey, thanks. The presentation was great. Question on fermentation. Did you use any microbial aids, or was it a natural fermentation after you added the cellulase?
Natural fermentation. Yeah, we didn't add anything.
So do you see like a dramatic change in properties X number of days later after doing that? Like were you measuring pH and stuff like that?
Yeah, we were measuring the pH. I don't have those numbers in front of me. It did change, but it started to smell different. So there was some like observational change.
Okay. Okay. And right now the intention for the fermentation product would be for cosmetics or nutraceuticals, or is it more for biostimulants?
The main kind of question around the fermentation was, does like an added step of fermentation help unlock the stuff that's in the kelp that the potential customers could be interested in the liquid fraction? So I think I think we'll get that answer when we get the results back of what was present in the liquid fraction, and if there were higher levels of that present in the fermented batch versus just the blanched or the non-treated batch. So, but yeah, generally the goal was broad market application, but with a focus on cosmetics and other ingredient suppliers.
All right. Thank you so much, everyone. I want
Thank
to be
you.
respectful of time, and I know that we're at the full hour and a half. Great presentations today, and I can send contact information if anybody would like. Jordan and I did have a couple overall broader questions that I think we'll just follow up with in an email. And again, yeah, thanks for your time. Exciting to see all the progress, and have a great rest of your day. Thank you.
Thanks, Alex.
Thank you, Alex.